Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. It has been a little bit of time since we have done a live stream, and it was necessary for me this week because last week at work was pretty busy for me at the hospital. And unfortunately, my video editor, had had some family health problems, so he is out as well. So I just wanted to make sure that we are moving forward with this oxidative stress series. And so I said, let's just do it live and deal with whatever mistakes I'm able to make during the video and just keep moving forward. So we have been talking, one second here. We have been talking about oxidative stress. And we last left off on this particular slide right here because really the last video that we did about how cancer protects itself against oxidative stress was mainly discussing one major system, and that was the glutathione system. And we were talking about how both glucose and glutamine are major contributors to that system, whether it be the creation of glutathione itself or by the recycling of glutathione through NADPH. So the reason I'm going to break this into a couple of different videos is because there are so many systems that are effectively being used against us. Because remember, these are our systems. These are our systems that we use to keep ourselves healthy, but they're being hijacked and they're being used against us in a way that protects themselves from oxidative stress. And what does that effectively mean? Well, it effectively means that it protects itself from radiation therapy. It protects itself against certain types of chemotherapy. It protects itself from our metabolic therapies. Because if we were to, let's say, employ an oxidative therapy like an IV vitamin C or an HBOT, but we don't take care of the antioxidant systems first, that's why this is a system. It's not one particular treatment. We're not going to have the level of success we'd like to have. Now, it's still, for me, a bit surprising when you actually look at vitamin C data, for example, of just adding it as a monotherapy to the standard of care, how effective it can be without dealing with these things. But it's still not going to have the effect that we're looking for without adding these things in first. So again, we hit glutathione, and today we're going to talk about the next system that is needing to be looked at from an antioxidant perspective, and that is the thioredoxin system. And this is a system that I'll be honest, I wasn't really familiar with until maybe about 4 or 5 years ago. And I definitely did not do any kind of deep dive on it until just recently when looking at how it protects cancer cells from oxidative stress and looking at it as a therapeutic target potentially to lower the shields, the internal shield system of cancer, and improve our chances of kind of destroying the cancer cell from an oxidative therapy perspective. So I took this paper not because it's quote unquote the best paper to discuss the thioredoxin system, but because this paper was published in August of 2025. This is, from a science perspective, this is really the cutting edge, the leading edge of where things are going. Okay. Now we're going to look at some other papers that I thought were better at looking at the thioredoxin system in particular. But this is a pretty good paper to explain kind of how medicine and science is figuring out how we should use these antioxidant systems against cancer. So it's titled Living on the Edge, ROS, Homeostasis in Cancer Cells, and its Potential as a Therapeutic Target. And it says targeting the antioxidant defense mechanisms of cancer cells has emerged as a promising therapeutic strategy. Inhibiting the glutathione system induces ferroptosis, a non-apoptotic form of cell death driven by lipid peroxidation with compounds like with Eferin A, that comes from ashwagandha, and ultramine, showing strong preclinical activity. Disrupting the TRX system by agents such as PX12 and dimethyl fumarate (DMF) impairs redox sensing survival signaling. TRX reductase inhibition by, I actually don't know how to say this, auranofin. We're going to talk about this compound during this video, but it's just a difficult word to say. Or mitomycin C further destabilizes redox balance, promoting mitochondrial dysfunction and apoptosis. SOD, superoxide dismutase 1 inhibitors, including ATN-224 and disulfiram, selectively enhance oxidative stress and tumor cells and are currently being tested in clinical trials. Mounting preclinical and clinical evidence supports redox modulation as a cancer-selective vulnerability. Pharmacologically, tipping the redox balance beyond the threshold of cellular tolerance offers a rational and potentially powerful approach to eliminate malignant cells while sparing healthy tissues, highlighting novel strategies for targeted cancer therapy at the interface of redox biology and oncology. And I thought that was a really nice summary and a true statement of how much this makes sense from a biological perspective and how powerful this could be. So it talks about superoxide dismutase and glutathione and other things, but today we're going to be talking about the thioredoxin system here. And again, here's superoxides, here's the catalase. We will talk a little bit about catalase, but I don't know if we'll be able to make it in this video. And then we've talked about glutathione, but this is another major antioxidant system that we're going to talk about today. So the paper that I decided to use as our kind of like major learning source for this particular topic was this paper that was published in September of 2024. And it's titled Exploring the Thioredoxin System as a Therapeutic Target in Cancer, Mechanisms and Implications. And I particularly highlighted one key component of this defense is the thioredoxin TRX system, which includes TRX, thioredoxin reductase, and NADPH. The thioredoxin system reverses oxidation of macromolecules and indirectly neutralizes ROS via pyroredoxin PRX. This dual function protects cells from damage accumulation and supports physiologic cell signaling. However, the TRX system also shields tumors from oxidative damage, aiding their survival. Due to elevated ROS levels from their metabolism, tumors often rely on the TRX system. In addition, the TRX system regulates critical pathways such as proliferation and neoangiogenesis, which tumors exploit to enhance growth and optimize nutrient and oxygen supply. And what we see here is basically just what that abstract is saying. It's the TRX system, although it is antioxidant defense, it protects DNA from damage. It helps promote metastatic spread. It helps proliferation, survival, et cetera, et cetera. So it's multifactorial of how it functions, but its main system or its main job is working as a powerful antioxidant system. One thing I'll mention here is that you see how it says here that it includes thioredoxin reductase, and NADPH. If you remember right from our last video, glucose through the PPP will contribute to the NADH that recycles glutathione, and it also recycles this thioredoxin system as well. So glucose is also important to this system as well as the glutathione system. So it says here, all cells have to balance the generation of ROS and their elimination by cellular antioxidant networks. The primary source of ROS is mitochondrial metabolism. Cancer cells have higher ROS levels than healthy cells due to uncontrolled proliferation and high metabolic rates. Elevated levels of ROS render cells vulnerable to oxidative stress. This weakness could be exploited to target and combat malignant cells. Therefore, there is a growing interest in modulating cellular redox equilibrium as a potential strategy for cancer treatment. It later says that the primary cellular ROS include superoxide anions, hydroxyl radicals, and hydrogen peroxide. In general, ROS production begins with electron transfer to oxygen, forming O2 or superoxide, which is highly reactive with a short half-life of 1 microsecond and limited diffusion through its membranes due to its charge. In aqueous environments, superoxide rapidly converts to hydrogen peroxide. Although not a radical, hydrogen peroxide is classified as an ROS. Compared to other ROS, it has a longer half-life, about 1 millisecond, and can diffuse through cell membranes. And it's rather specific, oxidating primarily free thiols, sulfur compounds. Hydrogen peroxide can be converted into water. This occurs through an enzyme such as catalase, which transforms